Okay, so my project is profiling the proteomic landscape of human breast cancer subtypes. So I wanted to start off with some breast cancer statistics. So breast cancer is the second leading cause of cancer death in women in the US. 1 in 8 women will develop breast cancer in her lifetime, and 85% of those women will have no family history of the disease. There are many methods of detection for breast cancer. The primary 3 are self-examination, breast exams, and mammograms. But the problem with these detection methods is that they're not able to detect breast cancer at an early stage or before the breast cancer metastasizes. So metastases, as you can see in the bottom right diagram, is when cells from the primary breast tumor break off and enter the bloodstream and then deposit on other organs in the body, which is what ultimately kills these patients. So the reason that it's so important to detect breast cancer at an early stage is that the earlier the treatment is implemented, the more effective it is and the better the prognosis is for the patient. So what we wanted to determine is whether we could detect breast cancer at an early stage using a simple method. And the best way to do that would be using a blood test. So blood tests for for cancer detection is not a novel idea. Many cancers are commonly diagnosed via blood tests, such as ovarian and pancreatic cancer, but breast cancer has an added complexity that there are many types of breast cancer. And so we not only wanted to determine whether we could see that a patient has breast cancer, but also say this is the specific type of breast cancer a patient has. So there are 6 main subtypes of breast cancer, but there are 3 that researchers tend to focus on. And we also focused on those 3, and those are boxed in red. So basal-like tumors, luminal A and luminal B are the 3 main subtypes. And basal-like tumors come from the outer mammary gland cells, whereas luminal tumors come from the inner mammary gland cells, which is just one of the many differences between these subtypes. But the primary difference between the subtypes is whether or not these cells have a specific set of receptors on the outside of them. The receptors in question are the estrogen receptor, progesterone receptor, and human epidermal growth factor 2 receptor. So we can say whether or not these receptors are present, that this is the way to classify these subtypes. So for instance, basal-like tumors don't have any of the receptors on the outside of the cells and therefore are commonly referred to as triple negative. The receptor classification is why we need to emphasize to detect breast cancer at early stages, because many of the drugs in current circulation for breast cancer are receptor targeted therapies, which means that many of them are only effective for luminal A and luminal B subtypes. And therefore, if a patient is diagnosed early with triple negative breast cancer, physicians can better understand which therapies they need to focus on instead of using therapies that they might guess would be effective without knowing the exact receptor content of those cells. Instead of using breast cancer patients, we used representative cell lines, which are the kind of the letters and numbers listed at the bottom of the screen. We used 8 cell lines for each subtype. So there are 3 cell lines per subtype, and these 8 in total were also compared to a normal cell line, which was used as our control group. Unfortunately, we did lose 1 cell line under the luminal A subtype during cell culture, but it wasn't a complete disaster, so we were able to continue with the study. So if ultimately our goal is to determine what type of subtype a breast cancer patient has, we first need to determine the molecular differences between the subtypes. And the best way to do that is to determine what the differences in signaling proteins are and in secreted proteins. So signaling proteins are the proteins inside the cells that allow the cell to interact with its environment. And that does not involve actually excreting proteins into the extracellular matrix. But secreted proteins is also allowing the cell to communicate with its environment by actually secreting proteins. So all of our cells are constantly secreting proteins, but when it's a tumor cell secreting proteins, these proteins become biomarkers, which means they become indicators of disease. So this is a simplistic version of kind of what we did in our lab to get to our analysis portion. We started off with cell culture. So cells are kept in dishes in nutrient-rich media, which allows them to grow and proliferate. And when the cells are at a certain confluency, we're able to extract those proteins, digest them into peptides, and then label those peptides with a specific ion. The ion then allows us to use mass spectrometry, which is a machine that then determines the intensity of the ion, which is directly correlated to the abundance of protein in that sample. So we can then say, okay, sample A had 20% more protein than sample B. proteomics and secretomics, which are labeled at the top of the pathway, determine which signaling or secreted proteins we're talking about. So proteomics is involved in signaling proteins, and to use the signaling proteins, we need to determine what is happening in the cell lysates. Secretomics, on the other hand, refers to the secreted proteins, and to determine the secreted proteins, we need to look at the cell culture media. So the conditioned media was also collected, and then we went through the same pathway that I just explained with protein extraction for the conditioned media as well. So there are many results that the mass spectrometer kind of spits out for analysis, but I wanted to focus on the most conclusive results for our study. So this is a PCA plot showing correlation between data points. So both the X and Y axes are able to show correlation based on how closely data points are clustered. So you can see that not only do the replicates within the same cell line cluster together, but also cell lines within a subtype cluster together, meaning that our sample preparation was accurate and we can see clear molecular differences between the subtypes. So each subtype is colored by blue or orange, and for instance, luminal A is orange here and luminal B is green. And you can see that the two luminal subtypes are more closely correlated because they cluster together more than, for instance, normal and triple negative, which is exactly what we would expect based on what we see in breast cancer patients. Another really representative figure would be this heat map. So to break it down a little bit, each of these lines is a protein. We found 693 proteins across the board. And what we can see is that some of the proteins, which are bright yellow, are upregulated, meaning that there's a higher abundance of that protein on average in that subtype compared to the other subtypes. Downregulated proteins are seen in dark blue. And so what we want to do is we want to come up here and say, okay, under the luminal A column, we're looking at this specific set of proteins, up and down regulated. Luminal B is this column, etc. So what we can do is say, okay, under the luminal A subtype, this specific set of proteins boxed in red is upregulated because it's bright yellow. So there's a higher abundance of those proteins under the luminal A subtype. We can then come to a different subtype, for instance, triple negative, and say that same set of proteins is downregulated in the triple negative subtype, which means that when a patient comes in and we see in that patient blood that there's a specific set of proteins, we can say with some conclusive evidence that that patient most likely has a luminal A subtype of breast cancer compared to triple negative because we see those proteins. This project has a lot of different future directions it could take. The first would probably be to finish our proteomic study. All of the results I showed you today are based on the secretome, and a lot of it has to do with the fact that during all this COVID-19 stuff, our lab shut down before I was able to analyze my proteomic study. So all of the sample preparation is done. The samples even got injected onto the mass spectrometer, but we weren't able to analyze them in time. Our second idea would be to repeat this with other representative cell lines. So we want to make sure that our results are conclusive for the subtypes and not just for the cell lines that we happen to use for the study. And lastly, we would want to ultimately see whether we can detect not only the subtype of breast cancer a patient has, but also the tumor stage that a patient has. So as you can see on the right, tumor stages are based on tumor size, lymph node status, and metastasis. And again, treatment is most effective if it's implemented early and as individually targeted as possible. So tumor stages would be really helpful to know as well. This study is by no means conclusive or comprehensive, but we just hope it's a jumping off point for a lot of future research. So just a quick thank you to everybody that helped me out, my PI, Andrew Amelie, my postdoc, Raghu, the rest of the Amelie lab for helping out with various analyses, the BU Department of Bio, which helped me complete this thesis, and of course, KHC for all their help and support and funding all the Ubers to take me to the lab at 5 in the morning to check on the cells. So thank you to everybody that helped with this project.